Hello everyone, good morning my dear friend, myself Renu Bharatkiram. I am learning B.sc second year. So today we are going to first online lecture. So let us start. DNA sequencing. So today we are going to know what are the DNA sequencing. DNA sequencing definition. DNA sequencing is the determination of the precise sequence of nucleotides in a sample of DNA. In other words, DNA sequencing is the process of determining the exact sequence of chemical building blocks called bases and abbreviated AT and C, ATC and G close bracket that make up the DNA. Method of DNA sequencing. Virus method used for sequencing are follows: Maxim and Gilbert technique. Second, dinucleotide method. Third, primer walking method. 4. Chromosome walking method. 5. Automated DNA sequencing. 6. Pyro sequencing. 7. DNA chips microarrays. DNA microarrays chips. It is the recently developed technique for DNA sequencing. The microarrays technique was first published in 1995 by M. Skechna and co-workers. A DNA microarray is collection of synthetic DNA probes attached to the design location or spot on solid surface. Microarray technique is similar to Northern blot, which is used to measure relative expression level of mRNA. Principle. DNA microarrays rely on the base pairing hybridization property of nucleic acid. Microarray chips. Microarray chip type. First, DNA chip probes cDNA in bracket 500 to 5000 bases long or oligonucleotide is immobilized to a glass surface. A robot is used to apply the spot. This technique was first developed at the Stanford University, California. Second, gain chip. Each gain has 16 to 20 pair of probes. Synthesis of the chip is pair. Each pair of probes has two sets of oligonucleotide. It is developed at Apermetric in bracket American company that manufacture DNA microarrays. Method. The major steps of DNA microarrays are follows: closing cell population. Second, conversion of mRNA into cDNA. Third, fluorescent labeling of cDNA. Fourth, hybridization of cDNA with DNA microarrays. Fifth, wash unhybridized cDNA off. 6. DNA scanning the hybridization arrays. Last point, interpreting the scanned image analysis. In figure 5.11, working of DNA chips. Application. DNA sequencing helps to understand the gain number of extra location and function of gain. Second point, the amino acid sequence of molecular weight of the proteins can be understood by DNA sequencing. Third point, it is helps of no regulation of gain expression for compression of the nucleotide sequence is newly identified gain of known sequence of gain help of corrected the virus gains sequence and produce of gain. 5. It helps to separate the genetic and environmental compounds of particular diseases. 6.8 It helps to know chromosomal structure of and organization. 7. It helps to know genes involved in complex traits and multi-gene diseases. 8. It helps to understand evolutionary conservation among organizations. 9. It helps to understand coordination of gene expression of protein synthesis and post-translation event. 10th, DNA sequencing has been used identification of gene responsible for the disease's cystic fibers. 11. Automated DNA sequencing has been successfully used to human genome project. 12. DNA chip is used to identification of genes responsible of inflammatory diseases. 13. DNA chip is used to identify the genome expression and hereditary diseases. Thank you.